of psychology. I'm in my 6th year of PhD, but I'm in the social psych area. And just this year, they decided that they're going to mix it up and assign TAs to different areas. So, yay! So I encourage you to give feedback about that to people in the department who have power. So I'm very sorry that you're stuck with me, but we will do our best today to get through everything. Let's make sure this is actually focused on the slides. So the way I was going to do this was to sort of go through, we can't spend a lot of time going through all the slides, but to go through sort of lecture by lecture, talk about the things, give brief points. I do, because I've seen his past exam now, have a bit of a sense of how he tests and things like that. So we can go through those, ask any questions about anything that is in these slides. So feel free to raise your hand at any point, ask anything, and then also, again, if you feel like you can explain this well, I would love your input on that as well. So. And of course, I also don't have the slides, so we have to scroll through it like a PDF. Fun. So this is from the first lecture. We'll probably spend the least time on this, but I think it's the most straightforward and the easiest to sort of look up if you need. I guess the last thing I want to say before we get started into content in terms of how to study, just based on looking at this past exam, what you really need to know is when you're thinking about the different studies that we looked at, you don't need to know the names of the authors. That can help you as a cue. He often puts those in the question, but you don't need to know who did this study. But if you do know that, it might help you. But he has questions like so and so study year where they did this, and this, and so it gives you enough information that you don't just need to know the name. That might help you if you're a person who is good at remembering names. That can be an extra cue. I'm not, but you don't need that to do well on the test. It didn't look like he previously used to test much on the reading. I don't know if that will be the same this time around, but there really wasn't much in past exams that looked like it was from reading. It looked like it was mostly from lectures. Any questions on sort of that kind of stuff? All right. So we started this quarter off with what is cognitive psychology, and he gave you a bunch of different quotes for. So make sure this is still going. Okay, beautiful. He gave you a bunch of different quotes from various famous people in cognitive psychology who give different definitions of what it is, but just to break it all down, they all really say almost the same thing, which is that cognitive science is the study of how the mind is organized and how that organization leads us to be able to have complex thoughts and to gain knowledge. So knowing that kind of general thing is going to be important. So these are all saying similar things, how we acquire and apply our knowledge and information. And we talked about why to study it. There's a lot of interesting reasons why to study it, but the most important, just the most basic one being, we want to know how the mind works. We want to know, is it fixed? Is it malleable? Is it both? How many parts are there in the mind? Are there these little modules, and how are they connected? And then this is also important because people are very interested in artificial intelligence and whether it can have consciousness, how our own consciousness arises, and how can we tell if something's conscious. So these are kind of all the big questions in cognitive psych. So also it has a lot of practical applications, and this sort of speaks to the interdisciplinary nature of cognitive psychology, where it interfaces with economics and social psychology, which is my area, and so in that sense we're interested in how people gain knowledge about groups and apply that in making judgments about groups, things like that. It's also useful in clinical psychology in the interest of mental health, in linguistics and how we construct language, and also in politics in terms of how we make decisions about who we vote for and things like that. So there's a lot of practical applications. This sort of reiterates some of the same things. We want to know about optimal mental health, how do we exercise the brain, interested in law, how we identify in eyewitness testimony, how accurate is that. You might have learned about that in other classes, but that we're often not that accurate, and cognitive psych has something to say about that. And then for politics, decision making and voting. There was an iClicker question. And then we got into the history of cognitive psych. And so sort of we can date this back to sort of the beginning of humans. We've always been interested in how our own minds work, but we've changed in how we think about that. So back in ancient Greece, Aristotle was one of the first to sort of think about the mind as actually being located in the body. Of course, he was wrong about where it was located. He located in the heart, it's important to know, and not in the brain, but it was a big step to say this is not a separate thing, it's not in the soul, it's actually in the body, that the contents of mental life have a basis in the body. Buddha and yogis also were thinking about how the mind works, and so just that this isn't just a new science. People have been thinking for the whole time that we could think. So just then sort of fast forwarding to more cognitive psych proper. The first psychology lab was started by Wilhelm Wundt. This is one name that you might want to know. And so that was the first psych lab where they're interested in actually introspecting, where you think about your own thinking, and so they're interested in studying cognitive psych in that way. Then we sort of go on to William James, who's probably the most famous psychologist, and he was interested in actually doing studies to get at this stuff, not just thinking about thinking, but actually doing empirical studies. Then we've got John Watson, and the rise of behaviorism. So that's this whole long era where they were really thinking about motivation and behavior as all being due to sort of the carrot and stick model, where you do things because you're avoiding a punishment or you're seeking a reward, but there really wasn't much interest in what's going on in the mind, in the brain. So that was sort of a black box where you have like a stimulus in and a behavior out, but they weren't really interested in what's going on in that middle piece, which is actually what's happening in the brain. Behaviorism and other key people in that are Skinner, probably the most famous one. And so yeah, they really just weren't interested in people's or rats' sensations and emotions and all the things that we think about now as being an integral part of psychology. They weren't really interested in that. Then there's Gestalt psychology, and so here they're interested in how, and we're going to kind of come back to this in other parts of the class, but that they're interested in how we get from these sort of features of an environment to being able to perceive a scene and a whole object. And so and that's a big part of how the mind works. How do we get from just like there's a line and a line and a line, and be able to see that as a table or something. So this is still an active type of theorizing now. And now we've sort of have had a revolution since the 1960s, in part because of gains in tech, but now we're interested in artificial intelligence. There's a lot of interest in human performance and maximizing that, in part because of World War II. We're interested in also the structure of language. Any questions? I'll pause there on sort of what we've just sort of rushed through. I know I'm going fast, but obviously there's a lot to cover. Yeah?
Are you supposed to memorize all that you went through?
So I can't say exactly what you need to know, but I think you need to know about the level that I'm talking at. So you don't need to know every single point, but you want to know sort of what's the general trajectory of this history. We started with not even locating the mind in the body, then we located the mind in the body, and we were interested in actually studying these things in an experimental way, and then behaviors and things like that. In the very back first, and then I'll get to you. Yeah. Yeah, you. Sorry.
Okay. So for Gestalt psychology, so when it says Gestalt equals figure, is it just saying like how we're able to organize those?
Yeah. Well, that's literally saying that that's the word for figure in German. And that's where it comes from, but the idea is, yeah, how do we get to be able to perceive a figure from just points of light and things like that? Does that answer that?
Yeah.
And then, yeah.
I was wondering, how detailed do you have to know the readings, or is it mainly on the slides?
I can't speak to how detailed you need to know the readings because I don't know, but I will say that at least in the past exam it didn't seem to be much, but I don't know what their reading was, so it's hard to say if maybe their reading just was more related to lecture. So I'm sorry, I can't really answer that adequately. Yeah.
Can you go back to William James and explain radical empiricism?
So I can't go too in depth in that because again I'm familiar with William James in a different context, but I'm thinking what he's going for there is that it was really the beginning of being interested in actually conducting studies and doing empirical psychology rather than being like, okay, well, how am I thinking about my thinking? And because this is juxtaposed as compared to in William Wundt's lab where they were doing introspection, where you're thinking about yourself. So that's like the key piece to know there. I don't know what's so radical about it, but I think it's radical in that it was departure from what was going on before. Got that today. Then now we're here on sort of the modern assumptions, which is that we can use the scientific method to fully characterize what is going on in the brain and what our mental processes are. So we haven't done that yet, but the assumption is that that is possible. That is the goal here, and that we can use algorithms or equations and models to actually think about these internal mental processes and to model them. And so then he gave the example with this graph, where this is where he was talking about if you have a series of numbers or letters, and so say you're always trying to identify, you're trying to find the T, and your first string of letters is just a T, so you're really fast to find that, then your next is like an L and a T, and so on. And the more letters or numbers that you add, the longer it takes you to find your target stimulus, even if it's in the same place every time. So even if it's in the third place in like a 3-digit string, and then in the third place in a six digit string, it shouldn't, in theory, take you longer to find it because it's always in the same place in that string, but it does take longer. And so this is speaking to that they were able to model this. So that's just an example of the kind of work that people do, where you take like a linear model and try to see, you know, is this a line or is this a curve? How does this actually line up with what's happening and what's the data? And then he goes into just some more on the modern approaches, which is experimental cognitive psychology. So kind of following the tradition of William James, where you actually do a study where you manipulate something. We talk a lot about these types of studies in this class. Then also computational, where you're really doing more of a simulation with a computer or something like that rather than actually studying individuals. And then also cognitive neuroscience, where you're getting more into the brain imaging and looking at how things are embodied. Yeah.
Do you think that a lot of questions you'll ask would probably end up like, this is an example of which one, like elemental, computational, or cognitive?
Yeah, I think it's going to be a lot of things like that, or as we get into more like specific theories and stuff, where he might say, you know, top-down processing looks like this, and he'll have a bunch of examples, and one of them is actually top-down, and one is bottom-up, and something else is like an unrelated type of thing that we talked about. But it seemed like he's pretty good at asking questions that aren't like a trick. He's very clear in the questions, at least in this past exam. I think he's going to use most of the questions for this new one. Yeah.
Are you able to explain top-down, bottom-up?
We'll get there, yeah. So then we went into sort of the anatomical landmarks of the brain. So I'm going to rush through this a bit because hopefully you've had some of this in other classes, and then again, this is stuff that's kind of just straight up memorization, which is unfortunate, but it is important to know. You'll use this in multiple classes throughout. So you want to know what a neuron is. It's the basic cellular building blocks of the brain. These are a bunch of different types. I wouldn't worry too much about which different type of neurons, just that they don't always look exactly the same, but they all have the same key components. So we've got the dendrites at the end, which are actually where the neurotransmitters, sorry, messed that up. But I think actually the key, the most important ones to know are the axon, where we're actually sending the action potential down that, which is the information that's in electrical form in the brain. So we use that to be able to communicate from one neuron to another using neurotransmitters, which are just chemical messengers in the brain that are released to communicate from neuron to neuron. And the synapse is the space where that communication occurs, where the two neurons meet, and there's a space there, and a neurotransmitter is released from one neuron, and it goes into the other, and that's how it gets the signal, and then it releases its own action potential, and it goes down the line and talks to another neuron, and so on and so forth. There are excitatory and inhibitory synapses, and that just means that ones that are excitatory, when they are activated, they lead the cell to fire more, and if they're inhibitory, they lead the cell to fire less. So both are going on. We talked about action potential, which is just the electrical impulse, which is carrying the information, and when it reaches the end, or the bouton, that's when it releases the neurotransmitter. So unfortunately, you're probably going to want to memorize all those pieces, but I recommend if the information here is not enough, there's lots of YouTube videos about neurons, or other articles that you can find. I definitely recommend just Googling stuff. That's how I learned like everything in graduate school, is by finding other resources. So that. So something I don't know too much about. I don't think you need to know too much detail, but just that there are limits on information processing, mainly in that there's membrane potential, and I think that's one of the limits on that. And I honestly don't know how that works, but I didn't see any questions on that, so fingers crossed. And then the key one that I think will come up is the rate of firing, is that neurons can only fire so often, and so that's going to limit how much and how fast information can be conveyed to other areas of the brain. That's really saying the same thing, just that, and then important is that individual neurons often respond to specific stimuli of different parts of different features in the environment, and that's kind of speaking to the on-off cells that we get to later when we talk about visual perception. But the key point there is that while they relate to certain stimuli, they can't explain cognition, because there's no single neuron that allows us to think. So we need to look at larger patterns of activation in the brain to get at that. So that's really the key point of this slide. And then more on sort of the anatomy of the brain. You want to know what this central nervous system is, being that it consists of the spinal cord and the brain. You want to know about the lower parts of the brain and the higher parts, so the lower parts being evolved earlier. We share those with almost all other animate life, and then our higher parts, which are in humans and other highly evolved species. And he goes more into that in terms of what the lower brain regions are. I didn't see too many questions on these, but I think it's good to know, just if you're going to be in psychology and you never know, sort of just the basics of what each of these more primitive areas do. So obviously breathing and swallowing are very key, so that's, we can't have evolved without that, so it's a very basic part of our brain, in which we need to just even be alive at all. So we just got an image of the brain with those different parts. Then going into these more recently evolved areas in this cerebral cortex, and just another important point is that in humans, we want to be able to pack as much of the cerebral cortex into our skull as possible, and so in order to make more surface area, it's all folded and wrinkly, and that's what the gyri and sulci are, and I would memorize those two. Yeah.
Do you know if he's gonna have diagrams of like the brain and the eye?
I don't know. I'm sorry. I don't know what he's going to do. I can't speak to that, so I can't answer questions about that. I can guess he might have a diagram, but he didn't have any images in his previous test. But he did say in class that he might do things like that, where he'd have a graph or an image and ask you a question about it. So I would be prepared for that. Just then the organization of the brain. So Brodmann identified 52 different regions that have different types of functions, and that these are sort of organized into 4 different lobes, and those are the parietal and the occipital and the temporal and the frontal. These are sort of intermediate areas, so they're not the most highly evolved and they're not the most basic, but the hippocampus, which is responsible for like learning and memory and emotion, and basal ganglia, which are responsible for basic motor control and also complex thought. And then you definitely want to know about the hemispheric specialization, or lateralization, it's also called, where there's different, the right and left hemispheres have different functions or specializations. So the left hemisphere being focused on language and analytic thought, and the right being focused on perceptual and spatial. So that's going to be key. And then the corpus callosum being the bundle of neurons that connects the two. And when that's cut, that's when you have a split brain patient. So he talked about some examples of that. And so if something's presented to the right side, you see it with the left hemisphere. And so if you have corpus callosum cut, then you would be able to say what that is. If it's presented to the other side and it's cut, you're not going to be able to identify that object. I mean, you'll be able to identify it, but you won't be able to say what it is. You might be able to make the motion for what it is. If it's a hammer, you might do this, but you're not going to be able to say that's a hammer. Whereas if it's on the other side, you'll be able to say that's a hammer. Is that there? And more evidence of this lateralization is seen in aphasia, and there's 2 different main types. In Broca's aphasia, it doesn't sound like they're hardly talking at all. They're just making short sounds like tan, 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 tan. They're trying to talk, but they can't actually generate words. And in Wernicke's aphasia, they sound as if they're saying meaningful stuff, but there's actually no content there. So it sounds like a sentence, but it doesn't actually make any sense. So if you didn't speak the language, you might think that they were actually talking in a meaningful way, but if you actually went in and tried to identify what they were saying, it would not have any meaning to it. Goes into more detail on where these are located, and so Broca's area is in the left frontal cortex, and like we said, they make short ungrammatical sentences, and in Wernicke's, it's in the left temporal lobe, so both on the left side of the brain, but in different lobes, and yeah, that leads to, it's hard for them to understand speech, and they also, they don't have any meaning in what they're saying. Questions? Yeah.
Someone who has Broca's aphasia, how would they talk today?
So someone who has Broca's aphasia would talk. The famous example is the guy they call Tan. He's going to say tan, 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 tan. And so he's trying to talk, but it's not coming out as any words, and there's no grammar to it either. It's just the same types of like repetitive sounds over and over again. When we talk about organization of the brain, we often talk also about topographic organization, which is just the idea that areas in the brain are organized spatially, so and that those map on to do the spatial organization of the actual world, and we get into this a lot more in the perception areas we talk about. And so this idea of like when you have, this is like from that study with cats, and the cats are looking at some type of image that's like this, and when they dissect their brain afterward and they dyed the something to make it show up which of the cells were activated, we see that it's actually mapping on in the same type of pattern. So the brain is organized where areas that are devoted to seeing a certain area of the physical world are next to each other if those areas of the physical world are also next to each other. And we also see the same type of topographic organization in the somatosensory and motor cortexes. If you took intro to psych, you remember the homunculus, where these things are, the tongue is near the nose and stuff like that. But importantly, they're not necessarily to scale. We have more area of the cortex is devoted to parts of our body that are more sensitive, or that we need to have more dexterity, and so the hand and the face and things like that. So then the last thing you talked about was methods, and you had some reading on that. And I didn't see too many questions on this, but that doesn't mean that might not change. But I would say don't worry too much about the specific details of these different types of imaging. Just know what their purpose is. So we've got the EEG, where you look at event-related potentials, and so it's basically you're looking at the activation of certain neurons or areas of the brain over time with the signal. I honestly don't know much about magnetoencephalography. Positron emission tomography is similar to fMRI, but you actually put something that's radioactive dye in the person to be able to see that. But it gets you the same idea of like you actually get to image the brain itself and just the electrical signals in the brain. This is some of what EEG looks like, and so you're looking at electrical signaling, and it looks different depending on what type of state people are in. I think that's about the level you need to know of that. I'm not sure what's going on here. Anybody got any ideas what the point of this one was? I don't remember him talking much about it, so I'm going to slide past it. Sorry.
Yeah, he didn't get to this part of the lecture.
Okay, then he said you're not responsible for anything. I missed one day while I was at a conference. I think this was the day I missed. So then we'll stop this one. Any other questions on what we've covered so far?
So if you didn't cover it in lecture, it's not
on the test. He said that's definitely true. That's one thing I can say with confidence. Go back, grab the next one.
On the EEG reading that he assigned, it went over some of that stuff.
Yeah, so I would definitely read that. And if I find an answer to how much you're being tested on the reading, we'll make an announcement. I'll try to get to the bottom of that. Yeah.
I think he said that we're responsible for everything on the lecture slides. I think he said that on Thursday after class.
Well, I thought the one thing he did say to me with certainty is that if he didn't cover it, he will remove it.
Okay.
So he means like everything on the lecture slides that he covered, I think. I'm sorry he's giving mixed information, but I think that if it wasn't covered, it's not going to be on the exam. Okay. Let's grab this next one. So now we're getting even more out of my wheelhouse here, but so now we're on to perception. The point of this slide was just that he's starting to motivate the concept that we have to sort of already have some top-down stuff going on where you have to sort of know what you're looking for in order to make sense of the visual world. And so with this, it looks like there's nothing, but once you know there's a cow, you're able to sort of look and find that. So he was just sort of setting that up, that we're lucky in the sense of like what we're gaining from all the visual processing is that we're actually able to make sense of splotches of light and dark in our world. And that's what he's showing here. just also that we can, our visual, we're able to get cues of movement even when there isn't actual movement. Now he's getting into more of the details of visual information processing, and so basically that we're beginning with the eye, and the eye is sensitive to light, so light passes through the lens into the vitreous humor, so that's this sort of area in here, so light's coming through the lens, it's in this vitreous humor, and then that image goes onto the back of the retina, but it's not totally sharp because it's gone through this sort of goopy liquid in the eye, and so one of the key things that's happening in our visual processing is for us to be able to sharpen and clarify that image. So you do want to know the main parts here, so the fovea, the vitreous humor, the retina, what those things are. He goes a little bit more into that here. Here are the key pieces, again, being so this is, that was sort of just the part due to the eye. Now we're getting into how that gets from the eye to the brain. So from the eye it goes through the optic chasm where the two cross, and this is why when we were talking about the split brain patients, if it's presented to the right visual field, trying to be the brain, then it goes to the left hemisphere because they cross there at the optic chasm. And then it goes to the lateral geniculate nucleus, and this is where those on-off cells were that we're going to get to in a little bit, through the superior colliculus and into the visual cortex, which is the part that's organized topographically. So this one looks a bit confusing, but the key point you want to know here is that the fovea is where we have the most clarity, and so you can see that here, it's a very small part of the visual field. It's these numbers 1, 2, 3, and 4, but when we look at how they're represented in the cortex, there's a lot of space that's devoted to that, and so that's why that's the area of the eye that allows us to get the most clarity, that we have the most area of the brain that's devoted to that relative to the size of the visual field. Questions there? Yeah?
So in the lecture, it's not on the slide, but you mentioned like ganglion cells and bipolar cells. You kind of drew out a picture of how they work, but it doesn't really say what they are, like how they function. Could you kind of like talk about that, or like do you know it?
I don't really know. I would say don't worry too much about that. I didn't see any questions like that, those more specifics, but I honestly don't. I tried to re-listen to that part, and without being able to see what he drew, it's really hard to cover that. So I would say don't worry too much. Yeah.
So I know you said to not worry about the slides you didn't cover. Does that also extend to like the readings?
As I've said a bunch of times, I have no idea if he's testing on the readings. I don't know. I just can't speak to that, unfortunately. I haven't gotten any information on that.
When I went to office hours today, he said like a little bit of it, like reducing the readings, but like how much is that? So
good to know. Yeah. So a little bit. I would say if you're worried about like how to study, focus on the lectures. Obviously, you should have already read the articles. If you haven't, I would definitely skim them, make some like bullet point notes, but don't get hung up on the details of it. If there's a term in there that we never even talked about in lecture, it's probably not something that you need to memorize. I would say that's a good way to approach that. Yeah.
Do you know how many questions there'll be?
I don't know.
Do you know if like he asks questions where like E would be, oh, it's also A and C? Do you know if he has?
It was a couple of those. There weren't too many of that.
Okay.
And where they were, it was pretty straightforward, where it wasn't too tricky. But again, I can't guarantee anything. I have no power. Yeah.
Are they all multiple choice?
Yeah. At least they were. So then here we're getting to these off cells, and really the key point here is that in the ganglion cells, so now we're in this area in the lateral geniculate nucleus. There's these on-off cells, and they're sensitive to light, and so just the key piece being like if it's an on-off cell, if light from the visual field is hitting the center of it, the cell will fire more. With an off-on cell, if it hits the center of the cell, will fire less. And then just to build this up, then you can also have bar detectors where they're lined up and they're longer, so you're able to detect not just the point of light, but the orientation of that bar. And then also edge detectors where you can see sort of the edge of a series of bars or something like that. So again, I wouldn't worry too much about all those differences, but I'm not totally sure. It's been very frustrating. And we move down to how we get depth. So we've seen how we get light. How do we know where in the visual field something is? And we have some different cues that we use for that. Texture gradient, stereopsis, and motion parallax. So we're going to go through each of those. This first being the texture gradient, which just means that when you're, for example, looking at like you're in a building or like you're looking at a brick road, and so when there's a pattern to that, and so bricks that are closer to you are going to appear larger, and they're going to get smaller and smaller as you go out, and that is a cue to the mind that there's distance happening there. So just like this, there's actually no, this is a 2D image, but because the spots are getting smaller, this looks like it's farther away. And same with this one. Then we've got stereopsis. This is a complicated image, but the main point here being that our eyes are offset from one another, and so because of that, we're getting slightly different images from each eye, and those allow us to be able to see in 3D because your brain reintegrates those two different images and tells you sort of where in space those are.
Excuse me?
Yeah.
Can you repeat that? What is stereopsis?
So stereopsis is a depth cue that we get that's due to the fact that our eyes are offset from one another, so you're getting slightly different images of the visual field. Like if you close one eye and the other, they're just offset a little bit, and your brain uses that to be able to know sort of how far apart things are, and it gives you cues of depth. And then we have motion parallax, and that's the effect that happens where I'm walking or I'm in a car, and objects that are closer to me, the angle of that it changes in terms of its movement is going to change faster for objects that are closer relative to objects that are further away. So like if I'm walking, my angle relative to people in the front row is going to change faster relative to my angle of people in the back row, and especially faster relative to my angle to the mountains or something back there. Those aren't going to even appear to move at all. But the angle of movement to things closer tells me if it's moving. If the angle is changing fast, then that object must be close. Questions on depth cues? Then we've got some more, these are all the principles of how we know that we've got, how do I say this? That we have this visual field, but how do we know that there are objects that are within this visual field? How do we know that this line goes with this object and not this other one? It's not just an amalgamation of random points and lines. How do we know what's a figure in that field? So we use a bunch of different principles. So we'll go through each of these. The principle of proximity is that we tend to group, if objects are close to one another, we see them as part of one type of figure. So like in A here, this is a bunch of lines that are just all spaced in different directions, but when you look at it, it looks like sort of this is one bar, this is another bar, this is another bar. Because they're in close proximity to one another, they appear to be sort of one object. Then the principle of similarity, where it sort of looks like this is one line and another line because the objects or the features within each of these lines are the same. So we tend to group objects that are similar together. Principle of good continuation is that we see this as being one line from A to B and one line from C to D. We don't think, oh, this is one piece here and another piece. We tend not to think about things like that. We want things to be a continuous line. We're more likely to perceive that. So we see this as a line and this as a line. And then the last one being principle of closure, where I don't know how to explain this one well, but that we basically, because this object closes, we see it as both being in front of the other one, because if it's a full object, it has to, I don't know how to explain it, it's just sort of an intuitive thing, yeah.
Even though something is occluded, you can make up a closure in your mind, because you know it should be.
Yeah, so we don't see this as a Pac-Man-like thing, we see it as a circle behind the others, just circle, like we can intuit or assume the fact that it's going on behind here. We don't see this as just this like moon or Pac-Man shaped thing and then a circle. So these are all different ways that we sort of go from basic features in the visual field to being able to perceive them to perceive them as objects. Yeah.
Can you repeat the principle of similarity one more time?
Yeah, so objects that are similar tend to be grouped together. So we sort of see these as going together and these as going together. We're not likely to sort of orient up down here because these are not similar objects. What he was going for with this slide, which he went over really quickly, was just that we don't equally attend to sort of all of the features, so they gave people this full thing, and then they asked them to say whether or not each of these different features was in that, and they were better at identifying sort of these ones that were closed or whole than ones that were sort of like, especially this shape, because we don't really, like, this sort of violates all of those different principles where we wouldn't, like, we don't see this as a feature, we see a triangle and maybe a right angle in another line. We're not really attending to this as a figure itself. Now we're on to template matching theory. Okay, so here this is the theory that how do we know, so how do we get from that retinal image to what we represent in the brain? And this is the theory that the image is transmitted as it is to the brain, and then we match that to different templates to know what that is. And so looking something like this, where you have some input with a bunch of different features, and we're trying to map that back onto the template that we have there. L doesn't quite match. Here it matches. This matches, but it's offset. So that's what they're going for there. And then the alternative theory to that is that we see stimuli as a combination of basic features, and we store those in our memory, in their parts. And so we don't store a full template for each A, but instead we're storing specific features of different stimuli. So that looks something more like. We're focused sort of more on the gestalt where we're looking at, we see we don't see each of these individual lines separately, but we see a combination of features. We recognize the H and we recognize the B here, even though there's actually a lot more images within this. There's also a 3, there's also a 4, but we see it sort of in the gestalt of, we see a full, we see, does anybody remember what the point of this slide was? I tried to re-listen to it and I could not glean that from there. I think what they're going for is that, I think this is saying that this theory doesn't quite work. Yeah, I'm honestly not sure. So this is something that, if we can use the discussion board to ask a question on that. I might put that in, or if anybody wants to add that to the discussion board, I think he's going to try to answer questions there that I don't know, because I did try to communicate with him that I wouldn't be able to answer all of your questions today, and I'm sorry about that. Yeah.
In class, he did say, like, this is kind of like an example against feature analysis, and then he said, because, like, once we know what our visual system is doing, we see complete packedness because we're not looking for the specific features anymore. So that was kind of how the HB thing went with.
Okay, so so this is a counter example to we're not just looking for simple features. We're looking, that we should, if we were just doing that, we would see an H and a B and a 3 and a 4, but we don't do that. We see features that are more meaningful to us, or that we don't identify all the features. We're more interested in the pattern. Thanks. Yeah.
Another resource said that this theory fails to account for the effects of contents or context. So like if we were looking for the 3 and 4, we could find it. And similarly, if we were looking for the H or the B, we could find it. So that's why this theory.
Okay, thanks. So now we're getting into visual pattern recognition. And so I would memorize these 3 stages, Biederman's 3 stages of how we recognize a visual pattern. So the first stage is that we see the world sort of as a bunch of cones and squares. So that this is just an example of like how that might look. And then the next, so that's the first step, is that we segment objects into a set of basic sub-objects. So instead of seeing like a full human, we see a bunch of sub-pieces to it. Same with the horse. And then step 2 is that once we've segmented them into these basic sub-objects, then we classify these into 36 basic categories, and these are the geons that he talked about. So this is step 2. We go from just squares and cylinders to a set of geons, and then he gave some examples of those. So there's cylinders and cones and pyramids, football, horn, like that. So these are like simple shapes. And then once we've recognized all of these pieces, then and their configuration, then we're able to recognize the object. So there, this is showing that there. And anybody remember the main point of this one? I didn't see this one much in the lecture, but I think what you do definitely need to know is just that this, I think, is support for this 3 sets of stages, that we can do this even without all of the information, that we can interpret this anyway. I don't think we got this far, did we? Okay, so stopping. And then go to the next one.
Yeah, he picked back up the next.
Then I think he went to attention and then he went back, right? Yeah, is that agree? Okay. Well, we'll go on to attention. This was just an orienting slide. He didn't really talk much about this. I think it's supposed to show an overview of all the different parts that we get to throughout the lecture. But one thing he talked a lot about is this serial bottleneck. And so this is the idea that we have a bottleneck in our ability to process different information from different streams. And so there's a bunch of different theories that speak to how that works, why that works, and that's what we're going to get into here. But that there's we aren't able to process everything in our world. That would be overloading. So there's 2 different sort of serial bottleneck theories. The first is that we select which to attend to before we recognize the pattern, and the other is that we select after we've recognized the pattern.
Excuse me?
Yeah.
You know the lecture that you had before?
Yeah.
And you said he stopped there. I think there was like a few slides that he went over specifically.
He went over some of the context part.
Let's see.
Scroll down. Yeah, like this grammar and then the word theory.
Yeah, so this is speaking very similarly to like the HB image, where this I think is speaking to the fact that we have this top-down processing where the context matters. And so when I say top-down, it means that we're using our higher functioning to inform how we see the visual world. So even though this is the same type of figure here, if it was bottom up, we should always see that as the same. But because we're smart and we use our cerebral cortex to help us process things, we use the context to help us know what this is supposed to be. And so because we can read, we say this is an H and this is an A, even though they look exactly the same. So that's an example of our top-down, that we use context to help us. If it were bottom-up, we would use just the features of the stimulus to tell us what that is. So this is just saying that same thing that we use. That's what the word superiority effect is that because we can read, we use the word to tell us what those what this specific configuration of 3 lines is supposed to stand for. I don't know what was going on here. I think this is just speaking to the same effect, but with our auditory system where you can drop out parts of a word and we just hear that anyway. We reinterpret that back in. You don't have to hear every single word or every single part of a word that someone's saying to be able to interpret that. This was the day I wasn't here, clearly. And then he got into this idea of change blindness, and this also speaks to our sort of context-dependent or top-down, is that we're not paying attention to every single feature of our visual world. He showed that video where within the course of like the 2-minute video, literally everything changed from the dead guy on the floor to being a totally different person, that because we're paying attention to whatever the main stimulus is or whatever's the most salient thing in the scene to be paying attention to, we miss other things there. That's then the most famous example of that is the study where they had this video of a bunch of people bouncing a basketball, and you're supposed to count how many times they pass it, and a person in a gorilla costume walks right through the middle, and people don't tend to see that because they're focused on something else. So our attention drives how we perceive things. What we're paying attention to influences what we see. Another example, this is a really fun study where they ask someone for directions, and then two people come by and they're holding a big board or plank, and then the person who they've asked the directions of is behind that, and as they pass, a totally different person is now there having the same conversation, and people will just carry on as if they haven't even noticed. So that's pretty amazing that we do that. And that was the end of that one. Okay. So now, back to attention. Any questions before I jump back over to this one? All right. Thanks for bearing with me, y'all. Okay. So now we're on this. So we talked about the dichotic listening task, and so this is an example of this serial bottleneck. So they have people listen to 2 different auditory stimuli in 2 different ears, and people are able to block out one and just listen to another, whichever you tell them to. If you say listen only to what's coming into your right ear, they can do that, and they can repeat back what's been said there. But they cannot repeat back what's happening in the other ear. So they're not able to do that when they're doing this. They cannot process both at once. But our attention can jump from one stream to the other, and this is what he calls the cocktail party effect, where you might be having a conversation with somebody and you hear your name or something else that draws your attention, and then you switch streams, and you might not hear what the person that you've been talking to is saying, and now you jump over to whoever said your name. So the idea of shadowing just refers to the fact that they are supposed to repeat back the words from one message only. They're shadowing what's happening in that, and so they don't remember much at all about the unattended message. They might be able to recognize whether the voice is male or female, or whether there's words or just noise, but they can't tell you what was being said, even if they repeat it multiple times. You might have the same thing over and over again in one ear, but you're not able to remember the content of that and repeat that back. Oh, here's the cocktail party effect. Yeah, so we automatically shift our attention. So then we get to filter theory, and so this is the theory that suggests that sensory information has to pass through some sort of bottleneck, at which point only some of that information is able to be processed out further, and that we select which message to attend to based on characteristics, whether it's the pitch of the voice, or whether they've said your name, or you've been told to listen to just one ear, but our attention drives which of the information makes it through that bottleneck. So then filter theory is that the idea is that how our attention shifts has to do with meaning also. So you can attend just to one ear, but if there are random words or lists, that we will sort of jump from stream to stream of message in order to make a coherent meaning. So one list coming to your left ear, for example, is saying dog, 6 fleas, and to your right ear it's saying 8 scratch 2, and your brain will try to make something meaningful out of that, and so it'll integrate those and come up with dog, scratch, fleas. And people are often not even aware that that came from 2 different ears, that they actually think that they're just listening to one, but because we're designed to find meaning and look for patterns, we integrate those. And in normal life, that would be excellent. You'd want to do that, but you don't usually have one thing coming in one ear and then the other. But that is able to show us how that bottleneck works, that our attention shifts to try to find meaningful content. Yeah.
I know the test hasn't been made yet, but like some of the theories have like the same name. Like there's two filter theory, like 2 experiments having to do with the filter theory, but they're like by different people. So like, do you think like you would generally give enough information, like where you'd be able to distinguish which one you're talking about?
Yeah. So these are 2 studies that are about the same theory as you said. So they're just, so he would say, for example, like Gray and Wedderburn's study, where they had people listen to 2 lists and with words, and some of them matched up to make a sentence from different ears. Like he would give you enough information to know which he was going for. But yes, they're both demonstrations of filter theory, kind of building on one another. First, this guy Broadbent is just saying that which we attend to depends on attention, and then this guy's saying that we try to build meaning out of that, and he's presenting a demonstration for that. That's a great question, though. So this is just showing that in an image that we just talked about. And then another example of filter theory with Treisman is another shadowing experiment. Oh, this is just summing up the 2 that sometimes we use a physical dimension of like it's coming to your right ear and sometimes coming to the left ear. You could divide it based on that, or you could base it on a semantic thing of this sentence makes sense together. And that's how we decide which stream to focus on. Then we've got the attenuation theory, is that the idea that certain messages would be weakened but not filtered out entirely because of their physical properties, so that they're still there, just not in their same exact form. And then late selection theory is the idea that the filter occurs sort of after the perceptual stimulus has already undergone some analysis for its content. This figure is attempting to show that. So in A, this is Treisman's attenuation theory, and so what's happening here is that there's a perceptual filter very early. So this is like stream 1 going to your left ear, for example, stream 2 going to your right, and here the idea is that the filter is happening early on, and you're really not even capturing much of this information. That's why the arrow goes from solid to dotted. You're still getting some of it in there, like maybe you know it's a woman's voice or something, but you're filtering out a lot of it. So if asked later to try to say what was happening in stream 2, you're not going to have much of that information. And then in B, this is Deutsch and Deutsch's late selection theory, and the key difference here is that the response filter is here rather than it being a perceptual filter. So it's not that you're losing information at the level of perception, but that you're losing it after you've analyzed it. Then there's a filter there that's basically making that less salient to you, and you're filtering it out, and so you're not attending to it as much. And so then the same result happens at the end where you have less information. Anybody want to add anything that they remember from lecture to that? I know that was a lot. Sum it up again. Here he's just saying you can use a metaphor for this, which is the spotlight metaphor, that we can move our attentional focus from place to place, but that we only have so much spotlight to give to something, but we can also narrow that spotlight to focus more in on something. Like you think about like a flashlight, you can have a wide angle where you can attend to a lot of things but not get as much information, or you can make it a tighter beam where you get more information, maximal processing on that part of your visual field. Now on to more visual attention stuff. So okay, so this is the study where they had a fixation point, and then so you're staring at that, and then a stimulus is presented just off to the side of that. So it's in your periphery, but not like in your periphery. It's still something that you can definitely see, but you're focused on the fixation point, and so people can see what's happening over there, but that they're faster at identifying that stimulus when it appears where they expect it to appear rather than where they don't expect it. And that just speaks to attention, that we're able to shift our attention without moving our eyes, but we're able to do that faster when we know where we're trying to shift that to, which makes sense. So this was a graph of that, where when you know where to expect it, you have a much faster reaction time to identify that a stimulus has appeared there. No expectation is somewhere in the middle, and when it's unexpected, it takes you much longer. Now I've got this, I don't know how to say this name, Neisier and Becklin, and they did a visual analog of the shadowing task. So the shadowing task being where you were having to repeat back one of the streams of information coming into your ear. So they did this with visual stuff, so people watched 2 different videotapes that were superimposed over each other, and they were instructed to pay attention to just one of those 2 films, and so they were able to monitor that and filter out the other, just like with when they were doing this with listening. So something like this, there's 2 different videos, but what they're looking at is those 2 on top of one another, so people were able to attend to one or the other, just like they could with the auditory information. Okay, and so this is showing a similar phenomenon, but here they, again, used overlapping stimuli, not in video form, but something like this, where there's a face superimposed over a house, and so people can attend to one or the other, and they can switch back and forth, and which you're attending to led to different activation in the brain. So when people were attending to the face, there's activation in the fusiform face area. When they're attending to the house, there's activation in the, I believe it's the temporal, or in the parahippocampal area. It's a parahippocampal place area. So that's just an area of the brain which is dedicated to recognizing objects and things like that, and as opposed to the fusiform face area, which is dedicated to recognizing faces. where's the, somewhere, I don't know, I think it was in a different lecture, but he had an image where it was showing neural activation, and so when there was when they were attending to the face, you were seeing a lot of activation in the fusiform face area and no activation in the parahippocampal. When they were looking at the house, you see activation in the parahippocampal and not in the fusiform, so really indicating that we're flipping back and forth. We're not attending to both of the things at the same time. They're looking at one or the other. And then we got into visual search, so how we see an array of objects and find stuff within that. And it can be relatively effortless when we're looking for something that's just like color, motion, size, or orientation, where it really sticks out. So it's really easy to find the red face here because they jump out to you. They're different in color. But what's more difficult is when there's a combination of features and you're trying to pull out one target that shares features with others. It doesn't have any, it's not entirely unique. So this is the example where they had a study where people were either trying to find the T in an array of I's and Y's, or within an array of I's and Z's. And because of the feature detection stuff that we talked about before, it's easier to identify it within these I's and Y's because the T has the line going this way and the line at the top, and there are no other stimuli in there that have that top line. So there's a unique feature there. But with the I's and the Z's, now we have both that top line in the Z and the vertical line with the I, and so it's harder to find. It takes longer to find the T in an array like this. So just showing that. There's that. And so this is just showing the reaction time for that. So with the T and the I and the Z, the more stimuli in that array, the longer it takes. Whereas it doesn't, you don't see as steep of a slope for that when you're looking for it in I's and Y's. Questions on that? Now we're at the Treisman and Galata study, and this is really just summing up, I think, this thing we just talked about, that things pop out when they're distinctive, and they're in an array of things that are different in the types of features that they have, and also that people are much more affected by the number of objects when there are these conjoint features. So that this is speaking to the fact that this is a much steeper slope, that the number of objects that you're adding is going to have a bigger effect on reaction times when there's these shared features than when there's not. Questions on that? Now we reach the binding problem. So now we have all these different features in the brain. How do we put these together to understand our visual field? Anybody remember what this means with the display info percept? Let's see if we can figure it out. I think this is, yeah, I'm sorry, I'm not sure what that was about. I'll have to go back and listen again.
It's talking about the slide that says visual attention up top a little bit.
Is it talking about, is it jumping back to this one?
So it's talking about that. So we scan it and we go through a process of picking the red vertical ones versus the horizontal green ones. It's just how we have a difference in what's being displayed and the info and stuff.
So just that there's different information in that. Mm-hmm. I think it's just saying there's lots of different features to attend to in the visual world. There's the color of it, whether it's vertical, whether it's horizontal. How do we integrate all of that into actually having an image of the visual field? So Treisman and Galata have the feature integration theory. So this is their answer to the binding problem. And so the idea is that people have to focus their attention on a stimulus, and then they can synthesize all of the different features into a pattern. So they need their attention to be able to do that, is what they're saying here. You can't just integrate the visual field without devoting actual attention to that stimulus. And so the study that was demonstrating that is that you're looking in the center of your visual field and there's a number or two numbers, and then off to the side there's a pink T or a yellow S or a blue N, and the idea is that people can pick up some of those features, but they have this illusory conjunction that happens where they see something off to the side and maybe it's a yellow T, and you ask them, was there a pink T, and they say yes. So they've picked up on that there's a T, but they're missing other features of that visual stimulus because they've been attending to something else in the center. And so that phenomenon where you report seeing a pink T when really it was a yellow T is called the illusory conjunction, meaning that you have this illusion that this other feature is attached to the one that you did see. Yeah, so people report seeing combinations of features that did not occur in that. Anybody remember the Treisman and Schmidt one? There weren't any questions previously on that, so hopefully there still won't be. And just that this is we know that this process is located in the parietal cortex. Important to know that this is the area where you're integrating what's happening in your visual world. So your occipital lobe or your visual cortex is where you're representing all of that information, but where you actually take that sort of just plot of what's happening in the world and you make that into a meaningful integrated image, that's happening in the parietal cortex. The point of this slide is just that our visual attention is spatially based. It's represented in like what we're seeing is tracking onto the visual world, and we know that because of what happens when people have visual neglect. There's hemispheric neglect that happens where people with brain damage to the parietal lobe ignore parts of the visual field. Actually, a friend of mine in psychology dressed up as this for Halloween one time, so he shaved only half his face and only had one of his arms through the sleeve, and they literally will only focus on or attend to things in the right part of the field. And so, like for example, if you give them like a plate of food, they'll eat everything on the right and they'll think they're done, and only if you rotate it will they then eat the other stuff. It's really fascinating how that works. And so this is just showing examples of that where you ask them to cross out all the O's and they think they're done, but they've only done the ones on the right. Okay, so now we're getting into more about what the parietal region does and how it integrates features. So the right parietal region is responsible for attending to the global features. This is going to be important, whereas the left parietal region is responsible for paying attention to the local, smaller features of that object. And so the example here is showing that. So A is the original stimulus that people are presented with, so either an M made up of little Z's, this triangle of little boxes, this complex, weird-looking thing. And so people who have right hemisphere damage in the parietal lobe, they're able to attend to the small features, but not the global thing. So they see that there's a bunch of Z's, but they have no conception of how those are oriented in space into a larger image. They see these little squares, but they're not orienting them into a triangle. And they're attending to these little details, but they don't have sort of the overall shape there. Whereas when the damage is to the left hemisphere of the parietal lobe, they see the global shape, but not the specific features. So they're seeing, I guess, the forest and not the trees, and these folks are seeing the trees and not the forest. Then getting into more on how we pay attention, this idea of object-based attention, that we tend to focus more on particular objects rather than regions of space, and this makes sense because in the world, like, things tend to operate as objects or figures or as a whole, not just like all these things in this region of space operating. And so the example of that was this, where they presented these sort of overlapping, they look like Band-Aids to me, to people, and people are much faster and the goal was that you have to identify whether there's the same number of bumps or different number of bumps on each of these stimuli. So this one would be, the answer would be yes, there's the same number. This one is also yes, there's the same number, but it will take people longer to identify this as having the same number than this one, because although these are close in space, they're not part of the same object, whereas this is on the same object. I had an iClicker question that was saying which one would you recognize faster. Say the same thing. Then also with this object-based attention, that there's this phenomenon that occurs called inhibition of return. So if we look at a certain region of space, we then find it harder to revert our attention back to that space. We're more likely to divert our attention to a new space, something that we haven't examined before. So Tipper and colleagues did a demonstration of this. It provides more evidence for this object-based attention, which is that, so you're looking at this this fixation point, and these points are moving around, and basically what happens is that, once you've looked at, for example, this point, then it's going to take you longer to look back at this one than to attend to one that you haven't looked at before. So just reiterating the point that we tend to attend to regions that we haven't already examined. It takes us longer to return back to rescan for something that we've already looked for over there. And normally that would be adaptive. In this context, it's a study, so they're trying to trick you. But this is interesting in how it shows how this inhibition happens, where without even any sort of conscious processing or anything, we're orienting to regions of space that we haven't yet devoted our attention to. That's the end of that one. More questions on this attention lecture? Yeah.
What did you say happened to the when the right hemisphere is damaged? Like,
so when the right parietal lobe is damaged, what happens is that they attend to the specific features or smaller features of some object, and not to its sort of global organization. So they see all the little Z's that are in this M, but they don't see that there's an M there. They're literally only seeing these small little features.
So if it was the left, and they see things as a whole, right?
Yeah. Well, the people with the hemisphere damage on the left, they see the whole but not the pieces, and then the right, they see the pieces, not the whole.
So it's like the left is like the global, and right is like the spatial.
Well, so it would say the left is responsible for seeing the small pieces, because when it's damaged, you don't, right? So this is saying, this is what people do when they have damage to the left hemisphere. So they're not able to see those small features. So the left hemisphere is responsible for seeing those small features, because when it's damaged, you don't. Does that make sense? And all this is occurring in the parietal lobe, because that's the area where we're integrating all of this information. Okay, so we've done now the first one, we've done perception, we've done attention. So now we're on the last one, which is this mental imagery. This is just orienting just what we're going to do that day, setting up the fact that we have a pretty strong ability as humans to mentally not just visualize, because it's not just visual stimuli, but what would be the right word for that altogether? Just, I guess we can call it imagery, even though they're not just visual images. So we're able to imagine, our childhood room with a lot of detail. Many people are able to imagine a song playing in their head. I can't reproduce any kind of song. I'm horrible at singing, but I'm able to hear that in my head. We're able to imagine these types of things, and that we're able to represent these in the brain, even when it's not actually occurring in the world, which is pretty cool to think about. what are mental images, that we use them all the time. People use them in meditation, trying to focus on something, or especially in like sports and things where you're trying to visualize your success, or visualize. He used the example of swimming, where you're sort of representing how long the pool is and your body's relation to that, and how many strokes it's going to take to reach the end. And so you can do all these types of mental imagery, which is pretty cool. And we can use this for, I can't remember what he said about dream recording, but the cool thing that he did talk about was that we're just starting to be able to sort of reconstruct what people are looking at or imagining using EEG and fMRI. This is just saying that what's remarkable about mental imagery is that the information is really like the same information as when you're actually perceiving something, but it's happening even though there is no external stimulus. And so it can happen with visual and auditory. He has in question marks, tactile and olfactory. So I wouldn't worry too much about that, but that it really happens a lot with visual and auditory imagery. And that's, if you like this kind of stuff, I definitely recommend Oliver Sacks's work. He's got a great book on hallucinations that I read recently, and we're talking about, because this mental imagery is so perceptual-like, that when people have some kind of seizure or damage, or when that's getting overactive, that's when hallucinations occur and things like that. And it really does feel like a real image because of our amazing capacity to do mental imagery. This slide is just saying that, just like with the fact that our we have different areas of the brain that are devoted to visual stimuli and to auditory stimuli, the same occurs when we do verbal imagery and visual imagery, that there's activation in different areas of the brain. I wouldn't worry about the specific regions. This is just showing again that the different areas are activated when you're doing different types of imagery. Okay, so this is a study that's demonstrating that these types of imagery are due to different types of processing. So I like this guy's name is Tanta. So he did a study where they presented people with either these different shapes in the geometric condition, or words naming these shapes. And they asked them to say whether they were the same or different, and they measured their reaction times. So this is you're supposed to try to match. So this is the thing you're presented with. You've got the triangle, circle, and a square, and you're trying to say if each of these different stimuli that you're being presented with is the same. So this one, yes, it's the same. This one's the same. This one's not because it's got that arrow. This one's not because it's got that arrow. And I think the key piece being that people are faster to identify when it's in the same configuration than when they're in a different configuration. And then you can also do this with words. So these are the answers. These are the same, different, different. And so the key outcome being that, so this is our reaction times here. And so when it's in the same configuration, we're really fast at identifying the geometric, if it's the same or different. If it's in a linear configuration, it takes us longer. So that's saying that it's going to take longer to identify this than this. Whereas with the words, the opposite's going to occur. We're used to reading things across a page, so if it's the same configuration, it can take us longer, even though it's the same. But if it's listed in like a linear thing, we're actually faster because that's how we're used to reading words. So it's just speaking to the fact that we process verbal and spatial information differently. Yeah.
I remember him saying that we need to remember the you can scroll up a few slides. like the different regions that are activated during mental imagery. So I don't get what the J and the R is.
J is the jingle.
J is the jingle, okay, and the I is and the R is the
The other thing I forget.
is the
Is it just
maybe the tracking from A to B. I'm sorry, I honestly don't.
He had a pen.
R is visual processing and J is verbal processing.
Right. So the verbal is happening in this parietal and temporal lobe, but I don't know how he would ask you to identify like this versus that, because we didn't learn the regions of the brain at that level of
You
detail.
said you should know like the back is the occipital, so you should know that's like for visual.
Yeah, I mean you definitely want to know that the occipital is for visual and the parietal is for integrating and temporal is for spatial.
Yeah.
But like this J versus that J, I don't think we didn't learn things at that level of detail. I definitely encourage for things that I'm not explaining well or you're not sure about, going back and listening to those parts of the lecture. I know he records them. When I was doing it, I put it on fast speed because you can speed those up and listen to him talk at a faster voice to kind of just like give yourself a refresher. But again, I'm sorry, I don't know more about that. Okay. So the implications of this. So this is where he was, I think, showing that video, and we can reconstruct what people are seeing from activation in the brain. It's not 100% clear because, of course, we have to integrate and process that image, but just represented in the visual cortex, we can see, what people are looking at when we can recreate that in fMRI, which is pretty astounding, and I think terrifying. I'm also an end of the world kind of person. Anybody remember the lucid dreaming example? I don't remember him talking about that. This is just summing that up, that different parts of the brain are involved in verbal and visual imagery. They represent different information processing, and the fact that we do that differently. Summing that up again. Oh, now we're getting to Shepard and Metzler's classic mental rotation stuff, and this you definitely want to know. So this is where you're trying to see if the second object, if you were able to rotate it, would it match the first? And so people are pretty good at doing that, but the key finding that they have is that the further you have to rotate it in space, even though you're imagining it, even though there's no reason that should take longer in terms of like physical distance because you're just imagining it, it takes people longer to say that they're the same or different if they have to rotate that further in their mind. So some of the images you only need to rotate a little bit to see if it's the same or not, and some of them you need to like flip all the way over. So for ones like that, it's going to take you longer to note whether it's the same or different. And so this was the importance of this, is that this really showed that the way that we imagine things is very much similar to how we actually manipulate things in space. You're literally imagining that thing turning, and that takes longer if you have to turn it further. Just saying that same thing, summing that up. Now we got to this crazy F study that we were talking about for a long time yesterday. And so this is where they're interested in how people scan mental images. And so they had people do it in 3 different ways: with pointing, with tapping with their left or right hand, or verbally saying yes or no. So the basic task is that you have this mental image of this block F, and then you're just watching this cursor sort of go along the edges, and you're saying at each point, is that the top or the bottom of the F? And if it is, you say yes. If it's not, you say no. So this corner is a yes, 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 and everything else is no. So that's the basic task, is just sort of tracking that and saying either by pointing, tapping, or saying yes or no, whether or not you're at the top or the bottom. And this is showing the same thing. This is, I think, is the pointing condition, and so this is a different way of asking about it. So instead of the cursor moving, at each point you say, is that a yes or a no? And so these are all correct. These would not be correct. This is like there's no yes in the middle of the F. There's no corner here, and these are also no. Okay, I tried to listen to this part again. I'm not quite clear on what's going on here. Does anybody remember this effect of the difference between the pointing, the tapping, and the vocal? Yeah.
Those are reaction times. Right?
Yeah, I know the reaction times, but do you remember why it took longer when they were pointing than when they were saying it out loud?
Because the spatial concept, you need to imagine in your brain and you need to think where to point.
Excellent. Yeah. So that what she just said so perfectly is that when you're pointing, that also takes having to orient yourself into space. That's a spatial thing you have to do, and you're also trying to hold in your mind a spatial configuration of things. So those are going to interfere with one another. So it's going to take longer to identify. Whereas if you're saying it out loud, now you're using a verbal thing and not a spatial thing. So they're not going to interfere to the same extent. Tapping is something in the middle. So then, to follow up on that, people were interested in, is it a visual conflict or a spatial conflict? Is it because in the one condition you're having to do two spatial things, or is it sort of which is accounting for that? So then they did this again, and this time, so in group 1, people are looking at, let me just make sure I get this right. They press it when there's a bright light, versus in the spatial condition, I don't remember the exact details of this study, but what they were going for is basically in one condition you just had to, you had the vision, you were representing it spatially in your mind, and then you had to indicate it using sort of a visual task where you press a button with using a light, and then in the other you're having to actually track that in space with a pointer. And the key conclusion from that was that when people had to simultaneously engage in conflicting scanning actions, it took them longer to do this mental scan. So when they had to track that pendulum in order to say yes or no, that made it more difficult to identify, because again, in the same way that she was saying before, that if you're trying to use the same areas of the brain, that it can conflict more. It causes interference in your mental scan. Yeah.
Do you need to know the experimenters?
You don't need to know their names or anything. He might have them in the question, but that won't be the only information that you get. He might say, Bradley and Lieberman did an experiment where they required subjects to do this, so he'll give you more information. If you like to remember things that way, like my old roommate, that's all he can remember is the names and the years. He would use that as a cue. I wouldn't. I would remember what happened in the studies. You definitely don't need to memorize those. I would know, the only names I would say to know are sort of just the key people in the history that we talked about in the beginning. So you want to know Aristotle and William James and Wundt, those key people. But in terms of every study person, no. Getting close here. Visual imagery. Okay, so this is where, again, now we're talking about visual imagery some more, and so we're doing this study where they're estimating the relative size of two animals from memory, and so their subjects take longer when the difference is small. That's just what that's showing here, that when there's a small difference in animal size, this is the estimated difference, our reaction times are higher. And same with the length of lines, not just with animals, but when the difference in length is great. Yeah, when the difference in length is great, we're much faster to say which is smaller or bigger. Okay, and so here what the key point here is that when this famous figure of it's either a duck or a rabbit, depending on how you see it, if you have people look at that and then hold it in their mind, they're not able to switch from one to the other. They're only able to visualize what they saw first. You can't say to them like, oh, well, was there possibly anything else that you saw in that? They won't be able to identify it, whereas if they're actually looking at that, they are able to switch back and forth. So I might have initially thought this was a duck, and if I was looking at it, you said, well, is there possibly some other kind of animal in there? I'd be like, oh yeah, maybe there's a rabbit. But if I was just holding it in my mind, I wouldn't be able to do that. So that suggests that when we're visualizing things, we're sort of holding it in this gestalt and not looking at the specific features as much anymore. We have this representation of the thing as a whole, and you're not very flexible in how you reinterpret that once you're in your mental imagery space. This is just saying that exact same thing in words. Brain areas. Just that the areas activated when you're imagining things correlate with the areas that are activated when you're perceiving them. And so this is what I was talking about before, where it was that image with the house and the face that were superimposed. Or if you gave people two different images, when they're looking at the face, you see activation in the fusiform face area. When you're looking at the house, you see activation in the parahippocampal region, and so those flip back and forth. So it's just saying that you get the same thing when you're imagining things as when you're actually visualizing them. You see similar activation. Then he did that trick question of the maps and looking at the map distortion. And what the key point is here is that we use larger objects to sort of give us information about where smaller things are oriented in space. So we have this idea that California is west of Nevada, and so even though there are certain cities like San Diego, which is further east than Reno, but because California is west, we tend to think of everything in California being more west than anything in Nevada. So these are just some images of that. We're likely to think that we won't think that Y is east of X because Alpha County, in which Y is located, is west of Beta County. Yeah?
Just going back to the brain area part, sorry.
Mm-hmm. No, yeah, no worries.
So when he was talking about the parahippocampal place area, was he saying that when you look at the face, this area is not engaged?
Yes.
Okay.
So you basically see that these are opposite. Like every time this is going up, the other one's going down.
Okay.
Like the bands are basically opposite. Yeah. So you're flip-flopping back and forth.
Okay.
This is just showing those things more. And that's the end of that. So that's all of the content from these. Yeah. Yeah. Go ahead.
I know you might not know what question he'd get out of this slide, or the ones with the map and everything, but do you know like what results you would get from that? Possibly?
From the map thing?
Yeah.
Oh, just the key point being that we're using larger objects to give us information about the locations of smaller things that are within it. So we tend to represent things at this more global level. Like we represent the spatial organization of the United States at the level of states, not of cities. And we get interference there where when there's situations like this where something that is within a county that's to the west is actually further east than the other, we tend to mess that up.
So it's like flipped too, so is that the same like, are you getting like the same like result from like if you were to flip it? Is that what you're saying?
If I were to flip which?
Is it flipped or is it just like in the incongruent one?
In this one?
Yeah.
Yeah, this is showing the same thing. It's just same thing can happen north-south. So here you're likely to think that Y is further south than X because Y is in Beta County, which is further south than Alpha County. So these are showing the exact same thing. They're just showing that it can happen east, west, north, south. It doesn't matter. Other questions?
Should we go over the visual scanning one more time?
Visual scan, yeah. What's that? This one, that's like F. Okay, so this one's confusing, but I think the key point here is that we can hold this F in our mental imagery. We represent it spatially, and so we tend to be faster at being able to identify where we're at in that mental image when we respond verbally than when we have to respond using some other type of spatial cue by pointing or tracking a light.
That's okay, so that's like spatial conflict then, is like.
Yeah, absolutely. Anything else? Well, I really appreciate all of your guys' patience. Next time he's going to give me more information ahead of time, hopefully. So thank you. And I will post this. I know it's not going to be like beautiful video, but at least you'll have something, and people who couldn't make it will have something.